Hi, my name is John Stukenborg from St. Jude Children's Research Hospital. We're so glad to be joined by you today for our St. Jude Talent Talk. We'll be discussing bioinformatics regarding the best of academia and industry. I want my guests to introduce themselves.
Hi, so my name is Avadhni Ramprasad. I'm a bioinformatics research scientist, and I work in the Department of Computational Biology. I was trained as a genetic epidemiologist.
Great.
My name is Jeremy Chase Crawford. I'm a bioinformatics research scientist in the Department of Immunology, and my PhD is in organismal and evolutionary biology.
And tell me a little bit about what you do here at St. Jude.
So the reason I came to St. Jude and what I do is to work on whole genome sequencing studies of two major projects. So one is the sickle cell disease genome project, and the second is the ALS project. So a lot of people don't realize that we study more than just pediatric cancer. Those are the kinds of projects that I work on.
What about you, Jeremy?
So I see my major role here as facilitating research by postdocs, grad students, and faculty members who are involved in cancer research, and as Avadhni said, lots of other types of research that people normally don't think about being affiliated with St. Jude. So people come to me when they are having trouble designing experiments or when they've generated a lot of data and they don't know how to analyze it, and my role is to solve those problems.
And what interested you specifically in a bioinformatics career?
So I was doing my PhD in the Bay Area at UC Berkeley right when the next generation sequencing technology boom really hit. And I was using these new technologies to investigate the evolution of the adaptive immune system. And I found that I really had a passion for figuring out how to push the newest technologies kind of past the bleeding edge to get the most data out of your samples, and then the statistics and the computation involved in getting the biological answers out of those data. And I just love it. So I kind of fell into it.
What about you, Avadhni?
Yeah, my story is a little bit different. So I was a faculty member before University of Miami, and partly I was working on ALS. I had put in a grant with Dr. Paul Taylor, and it got funded. However, that's not the reason why I came here. My husband was actually looking for opportunities at St. Jude, and I thought, let me look into it as well. And it just became a really nice coincidence that Paul Taylor was here, and I was coming here, and it was like an opportunity for me to kind of, I guess maybe expand like my forte into bioinformatics, because that's not how I was trained. I was trained as a genetic epidemiologist.
And was that an easy transition for you?
Yes and no. Of course, the hard part is always starting in a new place, but and also having been a faculty member before, I had to transition into a staff scientist position. But I can say that the training that I received in Gong Wu's group was really, really great, because he actually sort of mentored me through it. And so I feel like I've sort of retooled myself, and I'm able to now operate like as a bioinformatician as well as project management. So I guess in that way it was easier than I maybe anticipated.
Jeremy, what is your story of how you came to St. Jude?
So I grew up in northern Mississippi, and I grew up knowing about St. Jude. Everyone knows about St. Jude, but I had no idea how much research, basic biological research, is going on here, not just related to treating patients with cancer. So when I was finishing up my PhD, I had already decided that I didn't want to pursue a career in traditional kind of academia. I didn't want the headache of being a faculty member or a principal investigator. And so I started looking for opportunities to apply the skill set that I'd built up. And there was an opportunity here in the Department of Immunology, and I applied for it, and it just worked out to be a great fit. And so one of the things that I was kind of surprised by is how the breadth of the academic fields that the different bioinformaticians at St. Jude come from, and their histories. So like Avadhni said, she was a former faculty member. I didn't even do a postdoc. So regardless of where you are in your academic career, if you're interested in a job at St. Jude, I think there are opportunities here doing bioinformatics.
And what do each of you enjoy the most about working at St. Jude?
So a lot of things. I like being able to work in an environment where I don't have to worry about financing my research. So I feel like I am a scientist here. I get to operate as a scientist, not necessarily completely independently, but somewhat independently in terms of pitching ideas. But I don't have to worry about bringing in grants, and I don't have to worry about paying people. I can just sort of focus on science, and I really like that.
So I would echo what Avadni said, but I would just add that, you know, we have the brightest scientists in the world here at St. Jude, people at the top of their fields. And so I get so excited to work with these people. And I get bored if I do the same thing over and over. And I find that at St. Jude, I'm challenged every day with something new. And so that's really exciting to me. And despite the fact that I spend a lot of my time working with postdocs and graduate students who are pursuing, you know, their lines of research, I also have time for independent research, and that's really important to me.
And so you guys work in two different areas of St. Jude. Can you describe the structure of your team or your department as it relates to your specific roles?
Sure. So I'm in the Department of Immunology. I'm the only bioinformatics research scientist that's embedded within that department. We work sometimes with other bioinformaticians like Avadni, who are in the computational biology department. I work for about half of the PIs in the department, and then I work with others who come to me because they need help figuring something out. I kind of describe it as I'm a translator, and I operate kind of at the interface of the technology, the biology, in this case specifically immunology, but often broader than that, and the computational biology.
So I actually work with primarily two faculty members, the chair of the Department of Hematology, Mitch Weiss, and the chair of Cell and Molecular Biology, Paul Taylor. And I was brought here specifically to work on two projects. So the second project, the ALS project, is part of my research interest, more closely related to my research interest. So that's kind of consistent with me spending independent time on that research. But I also spend a sufficient amount of time just helping other projects and just sort of fielding questions and just generally helping our group, which is housed within the Department of Computational Biology. So the Department of Computational Biology has multiple different groups, and I'm part of the DNA group. There's also an RNA group, an epigenetic group, and also a clinical genomics group in that department.
And why is it structured that way from your perspective? How does that help our investigators and study teams?
Right, so the goal is that people like myself will be working in groups to try to assist other faculty members who have projects. They may not have bioinformatics expertise. In Jeremy's case, he's in a very unique situation because he's an embedded scientist within immunology, but there are other departments for which there aren't that kind of expertise that are there currently. So our goal is to sort of help them, to transition them, to sort of be that bridge, to teach them the tools that they need in order that they can do the bioinformatics work themselves, or we can do it for them. Yeah.
I think it's a great lead-in as we're talking today about, you know, the best of academia and industry and what St. Jude provides. Tell me, in your own words, how is St. Jude different from other institutions for bioinformatics research?
Well, this is kind of echoing what Avadni said earlier. I think the big thing is that at St. Jude, we really value great ideas. And so if you have an idea or someone comes up with an idea that, even if it's risky, if it has the potential to be transformational, they will come up with the funding to help you see that idea through. And so whether that's for equipment, for providing computational resources like our huge high-performance computing cluster, it's for reagents, it's for samples, there are really no limits. And so that really encourages people, I think, to think outside of the box, right? And it means that as a bioinformatician, I can work with scientists who are pushing the boundaries all the time. And I really love that.
So again, Avadni, from your perspective, how is St. Jude different from other institutions? You know, what makes our setup unique?
I think what Jeremy was saying is correct. I myself have not had one of those mind-blowing ideas, but I do believe that if I did, there is a venue for kind of pitching that idea. That's something very unique here. I think there's a lot of infrastructure and there's a lot of resources, and there's also a very strong academic culture. So we are in a hospital setting, and it is a non-for-profit, but this is a research institution at the end of the day as well. And I realized that when I came here. I didn't quite grasp that until coming here. Like the sort of the level of the speakers that come in for the Danny Thomas lectures, there's always something going on. There are a lot of like top-notch people that are coming here, and the scientists are doing really good job. I mean, it's a good place to be.
So thinking about your own work here and the time you've been at St. Jude, think about your most significant accomplishment, and what was that? Walk me through the scope of that project.
Do you want me to go first?
Sure.
Okay. So I have two things that I always kind of think about. I've been here for about 3 years, and the first thing is kind of revolving around teaching. So the graduate school is a new endeavor at St. Jude, and it was started about a couple of years ago when I started. And so even as a staff scientist, I was given the opportunity to actually design a bioinformatics course and to sort of direct that course with the help of other faculty members and staff members that actually taught in that course. So I feel like I'm still able to, even though I'm not a faculty member, I'm still able to contribute that kind of teaching and learning experience to students. And the second thing was, since I'm a little bit older than Jeremy, I've been sort of in the field a lot longer, where I've done like linkage analysis and other types of like array-based analyses and things like that. And so when I came here, it was really interesting because there was a large family from Australia that had ALL, leukemia, that was segregating, and nobody had been able to figure out how to pretty much figure out what's going on in that family. And so they had tried a lot of different things, but it wasn't until I came and used linkage analysis, which like is so antiquated but still useful, that we actually like narrowed down to a region, and then in that region I had to then apply bioinformatics tools to identify a copy number variant within that region that I wouldn't have been able to pick up with my past experience. You know what I mean? Like I was more of a statistician type. And so that was sort of like a really cool example of bridging the gap between genetic epidemiology and bioinformatics to kind of identify this causative mutation. So I think that's probably my proudest moment, I think.
That's great.
So far.
Awesome.
Yeah.
What about you, Jeremy?
I knew this question was coming. It's really difficult for me to pick, one or two things that I'm most proud of. I have to emphasize that almost every project that comes across my desk, I'm very enthusiastic about, and I mean that sincerely. We're not doing boring stuff here. There's always lots of interesting stuff going on. In the 2.5 years I've been here, I have a database of every project I work on, whether it's just helping write a grant or doing quick analyses or help carrying out the study. And I've worked on over 200 projects. And again, it's really a pleasure for me to feel like I'm helping these graduate students and these postdocs advance their careers and also advance our understanding of the underlying biology. If I had to, I could pick two. I can't narrow it down further than that. We also have an opportunity at St. Jude quite regularly to interact with some amazing medical fellows, right? So these are people who are trained in the clinic but are now doing research. And so one of the projects that we've been working on, we're about 1 year into a 2-year-long project studying pediatric acute respiratory distress syndrome. And this is an example of where the current standards in care haven't been able to really figure out what's going on with these kids. These are infants who have problems with their lungs. They come in, they're so sick that they have to be intubated. And we don't really know what's causing these issues. A lot of children die from this. And so we had an amazing clinical fellow who was interested in trying to tackle this problem. And so it involved a lot of planning, figuring out how we're going to get samples from the lungs of intubated infants, what we're going to do with them. And again, we're only halfway through this project, but I'm confident that we're going to figure this out. And I think we've already got some great leads. And that's really exciting to me, right? It's an example of where current practices are not cutting it, and we're taking on these challenging problems and we're going all in on it, right, to try to save kids' lives. And the other general example I would give is we're working a lot with CAR T-cell therapy, trying to engineer people's immune systems to attack their cancer and other diseases. And I've worked with some amazing clinical fellows in doing that, and it spans a lot of different aspects of bioinformatics, from gene expression to cell engineering to epigenetics, and so it always keeps me on my toes. And I also, it's exciting because I know this is the next big thing that's going to take us past these recurrence rates we have for pediatric cancer.
So my next question doesn't necessarily have to be related to your day-to-day work, but what is your most memorable moment here at St. Jude?
For me, it actually happened right before the Christmas holiday. I was in the cafeteria and I was getting breakfast. I don't know why I was here so early. And I just happened to be standing in line next to a man, like, you know, a man that was there with his child. And so I don't normally do this, but I struck up a conversation with him and we started chatting. And it turns out that it was his second time coming to St. Jude, and he had been there for like a full year before with his child. And it just kind of, you know, stuff like that happens a lot to all of us, I think, on a regular basis. Actually, in elevators, you can see people bringing like bags of McDonald's bags and, you know, just kind of wanting to tell their story. But it sounds a little bit cheesy maybe, but I think that really touched me and made me, you know, just kind of understand, again, you know, why St. Jude is a special place.
So for me, this isn't directly related to my work day, but there's a study ongoing at St. Jude that I'm not directly involved in any work capacity. It's called St. Jude Life. And the idea is we're trying, you know, so we have a lot of kids that come through here that we're able to cure cancer, but a lot of times they grow up to have other health problems in adulthood. And so they've built up this cohort of individuals that are already adults and we're lucky enough not to have cancer as children as a point of comparison, right? So kind of control points. And so I was able to participate in that as a control. And that was really important to me because my mother, my grandmother, and my sister are all cancer survivors. My sister had melanoma when she was 13. And so it felt like I'd kind of come full circle and that I was able to participate in research, even outside of the scope of my job, that might help someone one day.
And that really helps connect the dots, I think, to what we do at St. Jude. It's easy, you know, to stay focused on the work. It's very important work. But when we have, you know, moments like that, I think we're reminded of why we're here and why we chose to work at a place like St. Jude.
Definitely.
And along those lines, think about this. What is the one thing you would tell peers in your respective field why St. Jude is the premier place to work?
So, I mean, we've kind of talked about how there are just amazing resources and amazing opportunities here. One thing that I would like to emphasize is I think at a place like St. Jude, our mission is so important. There's really no place for ego here. It doesn't matter how famous you are, what your role is, if you're the chair of a department or you're a secretary. No one's time is more important than advancing the science so that we can find new applications to save people's lives. And so if you end up working at St. Jude, or if you're already at St. Jude, there are a lot of people who feel the same way that I do about that, and so you should seek those people out. And we're very collaborative. We love to work together to solve these big problems and help kids.
Yeah, that sounds good. So for me, I think, you know, so I think of St. Jude as like a big, small place. You know, it's like, people think of it as a small institution, but it's actually quite vast in terms of the diversity of sciences and the diversity of faculty and clinical operation that's going on here. So I think what I like about it is that although it's diverse and vast, you actually can get to know a lot of people pretty easily. So I feel like over the past 3 years, I actually know a lot more people on this campus than I did over the past 6 years when I was in Miami.
Wow, that's saying a lot.
Yeah, and I really like that because you have to, just the way that the buildings are set up and the way that you're interacting and collaborating, it's really easy to develop those connections. So that's something that I personally appreciated. I don't feel like I'm sitting in I'm not sitting in my own little office by myself all day. I'm actually like interacting and being part of like the greater scientific community.
Great. Yeah, no, those are awesome answers, and you know, just an opportunity for our audience to hear a little bit more about not just what you guys do, but how we connect it to the mission and why others should consider, you know, working here. And so with our audience, we'd like to field some questions for those that see the chat box there in our software. I have it pulled up on my iPad. You can type your questions in now, and I will be reviewing those, asking them as they come in. I do see some that are already in here, so I'm going to go ahead and start reading these off. So from Nadia, she says, hello, my background is in plant genomics and bioinformatics. Although the technologies and bioinformatics tools are used are the same between organisms, how difficult or easy is it for someone with non-human cancer genomics background to transition to work on cancer genomics?
So I think I can answer that. My PhD was, I focused on non-model murids and other rodent species from Argentina, Australia, North America. So I had no background in cancer genomics. I had no background formally in immunology. But I find that the people that I work with in immunology see that as a bonus. They see me as someone who's able to think outside the box and think about problems from a different perspective than someone who is trained as an immunologist. And so I say use that to your advantage. I think it's a big advantage.
Would you add anything to that, Avadni?
No, I think he hit it right on the nail.
And a general question of, is it hard to get a job when you cross fields like that? But I think you spoke to that, and while St. Jude is of course a competitive environment to get into, I think we need more people with that diverse background.
I agree.
Yes,
So, yeah.
yes.
Great. All right. Samarinda asks, what are the focused areas of research in bioinformatics in the coming decade?
Oh, wow.
That's a really
If we can predict the future.
striking question. So I think to your point, you could probably speak a little bit about CAR therapy and sort of the applications of that. I think that's a really huge area. From my standpoint, I think, I study populations of patients, and so I find that projects like St. Jude Life or the Sickle Genome Project are sort of hitting at what's coming in the future from a clinical translational standpoint. So they're not just collecting hundreds and thousands of samples and trying to sequence all of them, but they're actually following them prospectively to understand, are there any potential predictors that could be used for personalized genomics or personalized medicine, which is a really big term, and we're not there yet, but I think it's the right step forward. So I would say those are probably two that I can think of.
Yeah, I think personalized medicine is going to be a big one. Trained as an organismal biologist, when I got to St. Jude, I kind of had this mindset that, a cell is a cell, and the immunologists they will tell you that that's not the case, and I've learned otherwise. And so we are investing a lot in technology to assay, do a lot of single cell assays, whether it's gene expression, epigenetics, chromatin accessibility, and I think that's going to continue to be a huge thing. Every time we dig in deeper to epigenetics, to gene expression, we find that there are mechanisms that we didn't even know were working, right? And biology just gets more and more complex. So I don't foresee a future where we're not continuing to develop new techniques, new assays, and more need for bioinformaticians.
And I would, if I could just add with the techniques part, that the long read technology is definitely an area where people should start kind of understanding how to apply their tool set to analyzing that data, because that's definitely the way that things are moving in the future.
Absolutely. Now Hadi here is talking about, he works in machine and deep learning. Is there a place for someone like that at St. Jude?
Definitely.
Absolutely.
We have entire computational resources at St. Jude that are reserved explicitly for machine learning applications. It's something we're already investing in. It's something that, St. Jude, after I was here for a year, St. Jude paid for me to go take a summer course in machine learning, just so we could figure out, when to apply these tools that are available. There's absolutely a place for you here. Please apply.
There's also, we're also collaborating with Oak Ridge National, right? So they have huge resources and huge computational resources. And so it's there's no limitation in terms of the infrastructure that's here that would allow for that type of analysis to be done. And that's, because you can think about it, you can you can be in any university, reading up papers about machine learning, but you can't actually necessarily apply machine learning techniques if you don't have computational resources, and I think here we do. So I think it's a good place to be for that.
Good. So Jeremy, I know you had mentioned when we were talking earlier that there are opportunities to work on personal projects. And I know, Avadni, I think you brought a project from your previous role. Can you guys talk about how your roles are structured and that you're allowed time to work on personal projects like that?
So for me, I think part of coming here was the excitement of coming here was the ability to sort of transition from that faculty position into more of a staff scientist position, but to still retain some of the scientific sort of direction of the project. And so I function as what they refer to as a project manager here. I think, Jeremy, you probably function in similar roles, but so I manage like the scientific direction of aspects of that project that are related to my background. So I'm basically just working with groups and with collaboratively with other faculty members, and I'm designing, not experiments, but analytical experiments, let's say, from in a dry lab environment, and basically making sure that that runs smoothly. So it's the day-to-day operations, but also like learning about new technologies, learning about new methods, maybe even trying to develop new methods from a biostatistics point of view.
So, I mean, the main thing from my perspective is that I'm not micromanaged, which is great. I mean, when you're involved in lots of different projects and you work for multiple faculty members, if they try to micromanage you, it wouldn't work. So I have a lot of flexibility, which is really important to me, to kind of prioritize things and choose what project I'm working on at any given time. And that's really important because it allows me to kind of structure, okay, well, if I know multiple people kind of have the same need, even though they're asking very different biological questions, I can anticipate what we need to develop technology-wise to accommodate all of them, right? And so I can start building that. I can start putting that together so that it's available for the greater community here at St. Jude. About 20% of my time is reserved for independent research. Sometimes I dabble back in evolutionary biology because I miss it. Sometimes I give up that time because someone has come to me with a really interesting project and I want to help them see it through. So
great. So as we're working on these projects and others that are normally assigned to you, I know St. Jude is a resource-rich environment where we have access to some cutting-edge technology, specifically in bioinformatics in your respective areas. What is some of the technology that you guys got your hands on maybe before others, and talk through some of that?
So, do you want to
go ahead.
For me, it was working on the Sickle Cell Genome Project. So, Genome Project, as in the St. Jude Sickle Cell Genome Project. But one of the techniques that they were using was this idea of CRISPR. I think a lot of people will be familiar with that now. But when I first came, it was sort of like this idea that was out there, but I had never seen it like actually applied, and I didn't understand completely about ATAC-seq and about looking for epigenetic marks and going beyond just DNA sequence. I just didn't. That was not my training, and I didn't really understand that completely. So I felt like working on that project here really opened me up because I could see, from a statistical point of view, how you would identify those regions, from a biological point of view, how you would sort of interpret those, and then work with people to maybe downstream to design targets for actual CRISPR experiments. So I didn't do that obviously all myself. I was part of a team that was trying to do this for the project. So I got to experience and be part of that without having to, kind of learn everything myself, from the ground floor up. So,
wow, that's nice.
Yeah.
So the single cell technology is really big, and we spend a lot of time making sure that we are on the cutting edge of that. And whenever a new machine or a new product hits the market, we're investigating, is it going to help us do things faster, more efficiently, better, cheaper? So that's great. We have, I have to say, amazing resources for sequencing here. I've sequenced stuff all over the world, and it's never been as cheap as it is here. It's very fast, it's very efficient, and it's inexpensive. We're doing whole human genome for less than $900. So when you have resources like that, I mean, it seems like a minor detail, but that really facilitates research. Mm-hmm. The biggest thing I have to say is the samples that we have access to. St. Jude is involved not just in cancer. Every patient who comes in, their germline, their tumor, their whole genome, whole exome, RNA sequenced. We also have ongoing studies in HIV, in diabetes, in respiratory viruses. The availability of samples and the corresponding metadata is, I think, unparalleled, and it's just, you can't overstate the importance.
You're going to have to be our new spokesperson. That was great, Jeremy. And thank you both for joining us. I know we appreciated the questions that came in. I hope you guys learned something today. And you'll be able to review the recording that we're going to post on our website. But once again, just want to say thank you and enjoy the rest of your day.